Today we're going to learn about the stages of meiosis. We're going to start off with kind of a general overview and then we'll go into the more details. So remember, the job of meiosis is to do 2 rounds of cell division, so one cell becomes 4 cells. Now the purpose of that is to divide the chromosomes so the cell goes from being diploid and having 2 of each type of chromosome to becoming haploid and having only 1 type of chromosome. And that's going to, of each type of chromosome. That's going to be important because haploid cells need to come together to make a diploid offspring. So a haploid sperm and a haploid egg have to come together to make the zygote or the fertilized egg. All right, so let's walk through it. So this cell right here, we're starting off with our parent cell. This parent cell is a diploid cell. It has 2 chromosomes. The blue chromosome represents the chromosome from the dad, the paternal chromosome, whereas the red chromosome represents the maternal chromosome from the mom. Same size, same shape chromosomes. They just might have different codes, different versions of the same genes. So, for example, the dad's chromosome might have the gene for blonde hair and the mom's might have the gene for brown hair. Okay, so same type of chromosome, just slightly different codes. Now, right now, this parental cell, it has unreplicated chromosomes. So replication is going to need to occur during interphase. And during interphase, the chromosomes get copied, and now the chromosomes are made of 2 copies called sister chromatids. And if you remember, sister chromatids are identical DNA strands. So this diagram is showing the sister chromatids as kind of being like parallel lines. Oftentimes you'll see them as X's instead. So this cell could also be drawn like this, where we have 2 X-shaped chromosomes. That is fine as well. But in either case, we still have a diploid cell. We have pairs of chromosomes, right? And each, we still only have 2 chromosomes. They're just replicated chromosomes now. Now we're going to lead into meiosis 1. That is the first stage of meiosis. What's going to happen is those homologous pairs, those chromosomes, they line up together and they pair up together. So you can see the maternal chromosome is lined up next to the maternal chromosome. And when that happens, sometimes they can get tangled up in each other and they can cross over and swap parts. So let's go look at crossing over. We're going to look at crossing over in detail later on. So that's what happens in meiosis 1. Our homologous pairs pair up and cross over, and then they split. So meiosis 1's job is to divide homologous chromosomes. So we're dividing up those pairs. You no longer have a red and a blue chromosome, a mom and a dad chromosome. Instead, you have a blue or a red. But because of crossing over, you can see that there is kind of some swapping going on, and our red chromosome isn't totally red anymore, and our blue chromosome isn't totally blue anymore. All right, next up, after we divide the homologous chromosomes, meiosis 2 occurs, and its job is to divide the sister chromatids. So it's going to divide those identical chromosome copies. So you can see 1 sister chromatid going into each of those cells that's produced. So in the end, you get 4 total cells. So meiosis divides 1 cell twice to make 4 cells. It starts off with a diploid cell and it ends up with a haploid cell. So let's look at how that haploid situation happens. Okay, this cell that's starting off meiosis here, okay, it's a diploid cell. It's got 2 chromosomes, a pair of chromosomes. Because we split the homologous chromosomes in meiosis 1, these cells at the end of meiosis 1, each one of those is now haploid. It has a red copy or a blue copy. You no longer have a pair. So each of these is 1. So our 2 chromosomes were divided to form 2 cells with 1 chromosome each. The problem is we still have sister chromatids, so we need to divide those sister chromatid, those copies. So again, we divide one more time. Those gametes are going to be haploid, have 1 copy, a red or blue, or in some cases, a mix due to crossing over. Now, let's go into detail of the stages of meiosis. So meiosis starts off with meiosis 1. Meiosis 1 does prophase, metaphase, anaphase, and telophase. And many of those are similar to the steps that you learned about in mitosis, but there are a few little changes. So let's start off with number 1. Number 1 is prophase 1. Okay, it's the prophase, and it's occurring during meiosis 1, so it's prophase 1. Now, like mitosis, prophase has some jobs it has to do. Okay, there are 5 in total. 3 of them are very similar to what happens in mitosis. So you can see the spindle fiber is growing, our nuclear membrane is starting to break down, and the chromosomes have condensed. So remember, even though we didn't necessarily have it drawn this way in our other diagram, normally in interphase we have loose DNA. And then in prophase, that DNA condenses into X-shaped chromosomes. And in order for those X-shaped chromosomes to move, we have to make a spindle and get rid of our membrane. So those are the first 3 events of prophase 1. The next ones are a little bit different. Okay? Notice, we don't have the chromosomes just like floating around in the nucleus at random. They've started to pair up. So here we have 2 pairs. So in this case, we are a diploid cell. We got pairs of chromosomes, and there are 4 chromosomes. So it's a diploid cell. 2n equals 4. Okay? So the next thing you should write down is that there are pairs of homologous chromosomes. The homologous chromosomes pair. And then the last event that's unique to prophase is crossing over can occur. And that's because they're paired, they can swap parts. So let's go to the next picture and let's look at crossing over in detail. So here you can see, here's our paternal chromosome, here's our maternal chromosome. They have paired up in prophase 1, they overlap, and then they swap parts. Okay, so you can see in the end, our chromosomes are not all paternal or maternal. There's a little bit of mix of both. That's going to be an important idea because it increases the diversity of the gametes. And that means for more diverse offspring. Okay, after prophase 1 becomes metaphase 1. Metaphase 1, the chromosomes line up in the middle. But instead of lining up in the middle, again, we have them lining up as pairs. Okay? So we have pairs of homologous chromosomes lined up in the middle. And then the other thing that happens in metaphase 1 is we have spindle attaching to our centromeres so that you can separate those homologous pairs. The next stage is going to be anaphase 1. And in anaphase 1, this is where our dividing of chromosomes really occurs. So we are going to divide the homologous chromosomes. Okay, so what you can see is these homologous chromosomes are getting pulled to opposite sides of the cell. So that in the end, when we end up in telophase, you can see the cells that result, there's a large chromosome and a small chromosome. We no longer have a pair. There's not 2 large ones and 2 small ones. We have 1 of each. So we have divided those homologous pairs. So what this last stage is showing is it is showing telophase 1, and it happens at the same time as cytokinesis. Sorry if that runs off the screen. Okay, so in cytokinesis and telophase 1, there's a few things that are going to occur. Okay, the nucleus reforms, and notice we no longer have a spindle. Okay, and then in cytokinesis, that's when we're going to divide the cells in 2. So in this case, we got this animal cell, it's going to pinch in 2, and we're going to end up with 2 cells. So meiosis 1 makes 2 cells. And those two cells are haploid. So notice each of these cells, so if we can split it apart, each of these cells now has 2 chromosomes. We started with 4. Now we're down to 2, and we have 1 of each type. We don't have pairs anymore, so now we've created a haploid cell. Okay, so let's go on and let's look at what happens in meiosis 2. Okay, in meiosis 2, you're going to go through prophase, metaphase, anaphase, and telophase again. We just call these 2. So this is prophase 2. Okay, and you guys need to write down the major events of prophase 2. So that's going to be similar to what's happened before. Okay, we're going to have the nuclear membrane break down, our spindle's going to start to form, and our chromosomes are nice and condensed. Now, in metaphase 2, you can see that the chromosomes, again, are lined up in the middle, like in metaphase. And this time, they are individually lined up. So that's an important. Make sure you know that they're individually lined up. Okay, in the middle. And, again, the spindle attaches to the centromere. Okay, then comes anaphase. This is going to be anaphase 2. And this is when our main dividing happens. So in this case, you can see we're splitting up the chromosomes. So we're dividing these sister chromatids. Those chromosome copies that were made back in DNA replication. Okay, so here you can see we're splitting them apart. Okay, and then we end up again with telophase. So telophase 2, and again, cytokinesis happens at the same time. Okay, things that are going to occur in telophase and cytokinesis are nucleus. Nucleus is going to reform in our cells. There's no more spindle, okay? The cells are going to split in 2 because there's cytokinesis occurring. So in the end, let's look at our chromosome numbers and let's make sure we understand what's going on. So these started off as each of these being diploid cells. Okay, we had 2 chromosomes, a big one and a little one. As it goes through, each of these cells, so if we pinch them off, each of these cells still just have 2 chromosomes. Okay, so here you can see there's a chromosome here, a big one, and a chromosome here, a little one. So we still have 2 chromosomes. They're just unreplicated chromosomes, and that is the condition they need to be. And then these cells can go off and they can mature, and maybe they become sperm, and they can go get fertilized, or go fertilize an egg, I should say. All right, so that is the gist of meiosis. So let's just review what happens in mitosis versus meiosis. All right, so mitosis. Mitosis happens throughout the body, because remember, its job is to make new body cells for growth, repair, and asexual reproduction. But meiosis only occurs in reproductive organs, specifically the ovaries and the testes in humans. Okay, so meiosis is very limited in where it can occur. The purpose of mitosis, we're going to take one cell here, and we're going to divide it to make 2 identical cells. So the purpose is to create 2 identical body cells. Now, in meiosis, what we're going to result in is not 2 cells. We're going to make 4 cells, and they're going to be 4 genetically different cells. And the specific cells is that they're gametes. So they're eggs and sperm that are going to be used for sexual reproduction. Okay? So notice these chromosomes in these resulting cells, they're not always the same color. They could have been maternal versus paternal. There may have been crossing over that happened. So they're a nice mix. You got genetically different cells. Okay? So that was kind of the results. So the purpose for mitosis, you're going to have growth, repair, asexual reproduction, and meiosis is just for sexual reproduction. Now, the reason why you can get these changes is really because of what gets split. So notice we have, in mitosis, this cell is a diploid cell. We got 2 big ones, 2 little ones. So 2n equals 4. And then both of these cells that result are also diploid because they still have 4 chromosomes, big ones and little ones. They just are, and they're still in pairs, they just aren't replicated. Okay, meiosis, on the other hand, is going to take our diploid cell, as we saw, and because we divide homologous pairs in meiosis 1, and then we divide sister chromatids in meiosis 2, we have 2 types of chromosome divisions. In the end, these are going to be haploid cells with half. Okay, they used to have 4 chromosomes, now they just have 2, a big one and a little one. And which one they get is going to be very different depending on crossing over and how things line up. Okay, I hope that clarifies what happens in mitosis and meiosis. Be sure to ask me if you have any questions.